others, and also some empirical work we're doing with genome resequencing data and flycatchers. So before I jump into all this, I just want to sort of cover what is inbreeding, what is inbreeding depression, and why do we care about it. We can define inbreeding simply as mating between relatives. So an individual whose parents are related to any extent is inbred to some extent. So everyone here is inbred to some extent, particularly me from Tennessee.
So Marty's transition from us telling jokes about him being from Tennessee to him doing it.
Anyway, so the main effect of mating between relatives is to reduce offspring heterozygosity. So in individuals whose parents are more closely related tend to have lower heterozygosity on average across the genome. And this occurs because of some loci in the genome of an inbred individual are identical by descent. And that occurs when an individual inherits two identical copies of a chromosome segment which arise from a common ancestor of the parents. So this individual could carry identical by descent loci arising from this female or this male. I should say also, if anyone just wants to stop me and ask a question or discuss something, please feel free to do that. And when an individual is inbred and carries identical by descent loci across the genome, that individual is more likely to be homozygous at loci carrying deleterious recessive alleles. And what this creates is this negative relationship between the fitness of an offspring and the parental kinship or the relatedness of the parents. So individuals whose parents are more closely related tend to have reduced fitness, and we call this phenomenon inbreeding depression. So the total effects of inbreeding on individual fitness can sort of scale up to the population level to reduce the population growth rate, and that's why all of us in this room should care about it. And there are I'll just show you a couple recent examples of studies of inbreeding depression in natural populations. This was from Joe Hoffman's group in studying harbor seals, where they found that both longevity and resistance to lungworms was associated with genome-wide heterozygosity measured with a relatively large panel of SNPs, more than 13,000 SNPs. So individuals who were sampled when they were old, or lived until they were old, tended to have higher heterozygosity than individuals who died young. And individuals who were not infected with lungworms tended to have higher heterozygosity than individuals who were infected with lungworms, suggesting there's an effect of inbreeding on both of these variables. This is a recent study from Josephine Pemberton's group, where they looked at effects of inbreeding on lifetime breeding success and some other fitness components. So that we have on the y-axis here lifetime breeding success, so number of individuals per offspring produced as a function of a genome-wide measure of individual inbreeding. This is specifically the diagonal elements of a genomic relatedness matrix. And more highly inbred individuals tended to have much lower lifetime breeding success. So this is apparent, really strong inbreeding depression. So why do we care about inbreeding depression in various fields? So in evolutionary biology, we should care about it because inbreeding affects individual fitness, and we care about that in evolution. And specifically, inbreeding depression is thought to strongly affect the evolution of mating systems and the evolution of behaviors to avoid inbreeding and dispersal. And in medicine, we care about inbreeding depression because we're interested in the basis of the reason for recessive diseases. And in conservation biology, we really care about inbreeding depression for the management of small captive populations, which tend to be very highly inbred, and also for quantifying and managing the viability of wild populations. So what are some of the outstanding questions, the important questions in the field right now? One is, what is the genetic basis of inbreeding depression? How many loci are involved? What's the distribution of effect sizes? This is really important for understanding purging and how likely deleterious recessive alleles are likely to be purged from populations. Which fitness effects are most frequently depressed by inbreeding depression, and a related question is, what are the effects of inbreeding depression on population growth rate? Sometimes inbreeding depression or inbreeding could affect fitness components that have very little effect on population growth rate. For example, juvenile survival in ungulates. Whereas it could also affect fitness components that strongly affect the population growth rate. It's important to understand what traits are subjected to inbreeding depression and how strongly those traits affect population growth. And addressing any of these questions really requires us to be able to measure individual inbreeding in some precise way. So before I talk about how we measure individual inbreeding, I want to talk specifically about the genomic effects of inbreeding. So how does inbreeding affect the genome? I've already said that the major effect of inbreeding on the genome is that individuals who have more closely related parents have lower heterozygosity. Why does this happen? And to illustrate this, Fred showed a similar figure the other day, but this is a different one from simulated data. This is a pedigree of two full sibs of parents whose parents are sibs. So we have two inbred brothers here. We have sort of the founders of the pedigree at the top, the grandparents. Each of them has a unique copy of a single chromosome. These are mapped by the different colors. And if you follow the chromosomes through the Mendelian inheritance and recombination events through the pedigree, you can see where recombination events occurred by the boundaries between different colors. If you look at the chromosomes of these inbred individuals, the inbred brothers, each of them carries a few, one in this case and two in this case, segments where the individual carries two exact copies of the chromosome segment which originated in one of the grandparents' chromosome copies. So if you look at this chromosome pair from this brother in particular, and we look at a map of heterozygosity at simulated SNPs across the genome in 500 kb windows, I think, you can see that there are these two chunks of the chromosome which are completely homozygous. So that's what we see in the genomes of inbred individuals, is these big chunks of chromosomes that have no heterozygous position. So we have this parameter F. So what this is sort of what I consider being realized or true individual inbreeding, and that is the proportion of the genome that's found within identical by descent chromosome segments. So if we can identify these segments in individuals, we can sum up their lengths and divide by the genome size, and that's the true description of an individual's inbreeding level. That parameter is called F. So we have these identical by descent chromosome segments, and they have a distribution of lengths. And the expected length of an identical by descent segment is determined by the number of generations separating an inbred individual from the common ancestors of its parents. So if you have a common ancestor of the parents that's a few number of generations ago, there are very few meioses separating the inbred individual from the common ancestor, so we tend to see very long identical by descent segments. With an individual whose parents are full sibs, you can see entire chromosomes. They're completely homozygous because there have been no recombination events in either genealogy between the inbred individual and the grandparent. Whereas if you have a common ancestor that's many generations ago, you tend to see very short identical by descent segments because there have been many meioses and many chances for recombination events to break up these inherited segments into shorter chunks. So these are some simulated data that I did. I simulated inbred individuals whose common ancestors were 2 generations back, 5 generations back, and 10 generations back. So as you increase the number of generations ago the common ancestors of the parents were, you see this shift to the left in this distribution of IBD segment lengths. So these figures should make it really clear that the inheritance of these IBD segments is an extremely stochastic process. So these are 2 individuals with the same parents. They have the same exact pedigree, but they have different lengths of IBD segments and a different proportion of the genome that's identical by descent. So you can have individuals with the same pedigree that have vastly different realized levels of genomic inbreeding. And the standard deviation, or the variance in F among individuals with the same pedigree, is determined by the number of chromosomes in the genome, the map length of the genome. Basically what these two things do is determine the number of the frequency with which recombination happens across the genome. And also the number of generations separating the inbred individual from its common ancestors. Just to illustrate this, this is from theoretical work, some old theoretical work by Franklin. If you look at the expected standard deviation, the standard deviation in F among individuals with the same pedigree, in this instance, after one generation of selfing versus the number of chromosomes, as you increase the number of chromosomes, the standard deviation goes down. And as you increase the map length of the genome, these are arranged in this order. So if you increase the map length of the genome, you also get a lower standard deviation in F among these offspring produced by selfing. So this is another illustration of this. So there's this, a lot of you have probably heard of this population of wolves in Isle Royale, which is in Lake Superior. And right now, this picture was taken last year. These are the only two adult wolves left on the island. The one on the left here is their pup, who's thought to have died last year. This is the pedigree of that pup, and its pedigree inbreeding coefficient. So the proportion of the genome predicted to be IBD, given the pedigree. And Phil Hedrick and I used some simulations to determine how precisely the pedigree inbreeding coefficient can predict the realized genomic inbreeding of an offspring of the two remaining wolves on Isle Royale. We use genomic simulations, assuming the linkage map of the dog, so basically we know the map length of every chromosome of the dog genome. We assume it has the same map as the wolf. And we simulated the whole pedigree. We had to make the assumption that the pedigree founders were unrelated, because we don't know how related they were, and we don't have genomic data from them. And then we calculated the realized level of genomic inbreeding among 5,000 simulated offspring. And the mean of this distribution is exactly what we would expect, 0.438. It has a pretty broad distribution of actual values, ranging from around 0.2 up to 0.7, with an expectation of 0.438. There's a fair amount of variation in precision in how well we can predict realized genomic inbreeding given the pedigree in this population, for offspring of these individuals. It's just one example. So when we study inbreeding depression, how do we measure it? So I've already talked a little bit about the pedigree inbreeding coefficient, and I'll describe it in detail in a moment. This is the classical approach. It's the classical way of defining individual inbreeding is by the pedigree inbreeding coefficient. And we can also use genetic markers to measure individual inbreeding because the signature of inbreeding on the genome is reduced heterozygosity. And this can be done with both unmapped markers and with mapped markers. Unmapped markers have been used for a long time, since the 1970s, using just a few allozymes, and then more recently we're using up to hundreds and thousands of SNPs, unmapped loci. And then with mapped markers, we can explicitly try to identify these identical by descent segments by identifying runs of homozygosity in the genome, so these long tracks of homozygous genotypes at mapped loci. So what is the pedigree inbreeding coefficient? So this is a classical measure of individual inbreeding. Sulbright was the one who did this first. So if we know an individual's pedigree, we can calculate the expected proportion of the genome that's identical by descent. Wright's parents were first cousins, so he estimated his own individual inbreeding coefficient at 0.0625. Always wondered if that's why he was interested in inbreeding. So some important characteristics of the pedigree inbreeding coefficient, which we've known for a long time, is that it does not account for inbreeding into distant ancestors. So this pedigree inbreeding coefficient here assumes that these two individuals are both non-inbred themselves and that they're unrelated. So essentially that there are no inbreeding loops that extend beyond this generation in this pedigree. And it also doesn't account for this sort of stochastic effects of the combination on and Mendelian inheritance on the imprecision of the pedigree breed coefficient. So when we can use unmapped markers, and basically this is an approach of using the proportion type loci that are heterozygous as a measure of individual inbreeding. We can do this because individuals who are more inbred have lower heterozygosity in the genome. And this is known to be highly imprecise when relatively few loci are used. If you only use a dozen microsatellites or 3 allozymes or something like that to estimate multiple locus heterozygosity, the sampling error is enormous and the estimates are incredibly imprecise. When we have mapped loci, we can try to explicitly identify these runs of homozygosity which are indicative of the presence of IBD chromosome segments. This is just sort of a cartoon of a chromosome with a bunch of mapped SNPs where ones indicate heterozygous SNPs and periods indicate homozygous SNPs. So basically what we do in these sorts of analyses is to scan the genome in some way, and there are several different algorithms we can use to do this, one of which we'll use in a little while. So we look for parts of the genome where we see very few or no heterozygous positions. And a complicating factor is that we often see some heterozygous positions within truly identical biosynth segments because of sequencing error and the occasional mutation. So we have to account for this in analyses. Some of the approaches that have been used are quite variable. One is implemented, a simple approach is implemented in the program Plink, which you guys will play with later or tonight. Basically, it scans the genome using sort of user-defined criteria for what we think a run of homozygosity should look like. And I'll describe those parameters in detail later. We can use a PSMC model. It's an approach that's often used to estimate deep historically effective population size. Basically, what the model does is estimate the time to the most recent common ancestor for chunks of the genome. We can use it to identify parts of the genome where we think that 2 copies coalesce at a very recent common ancestor. And this is the approach that I use most often. It's for sort of chunk windows of SNPs in the genome. We calculate a log odds ratio, basically the proportion of the probability of the genotypes assuming they're identical by descent, and the probability of the genotypes assuming non-IBD. And when we see large values for that, it's indicative of the segment being identical by descent. So what are some advantages of using marker-based measures of F? You don't need a pedigree. You don't need generations after generations of parentage information to build. They're not necessary in order to use marker-based measures. And they also capture variation in breeding due to distant common ancestors of parents. And they also capture variation in inbreeding that arises because of linkage. And we can also use markers, in some cases, to identify loci that contribute to inbreeding depression, so we can learn about what the genetic basis of inbreeding depression is. So these are some data from a study in humans, where they estimated the proportion of the genome that was in runs of homozygosity using molecular data, and plotted this against the pedigree inbreeding coefficient. And the fact that the intercept is above 0 illustrates that this measure is capturing inbreeding that's deep in history. It's not captured by the pedigree in this case. So a major disadvantage in marker-based measures is that you have imprecision sampling error arising when you have relatively few markers used.
What is relatively few markers?
It varies a lot. Yeah, 100 is always relatively few. 1,000 is maybe relatively few. A few allozymes is way too few. I'll talk more about that. So these genomic approaches, like FRO, have been used pretty widely in humans, but they're also starting to be used in some natural populations. These are some data that Fred showed the other day from the study of mammoths. So on the y-axis here, we used a PSMC model to estimate the TMRCA, the time to most recent common ancestor, across the genome. So basically what they did was they had sequence data that they aligned to the elephant genome. So they're assuming syncing with the elephant. So where you have parts of the genome where the TMRCA is very low, so basically you have the two homologous copies of the genome coalescing with a very recent common ancestor, you can call them as being identical by descent, with the IBD tracks identified and marked by these gray bars here. And as Fred said, this individual is sampled from a remnant population on an island. This individual is sampled from the mainland, non-remnant population. This individual is highly inbred, as we would expect. This approach has also been used in Soay sheep recently, using a 50K SNP chip in Soay sheep. That's a relatively small number of markers to try to do something like this. Identifying runs of homozygosity is pretty imprecise unless you have a really huge number of markers, hundreds of thousands of loci or more. This study in gorillas, published in Science recently, looked at runs of homozygosity from 13 individuals using genome resequencing data. So, sort of recognition of the sort of more recent recognition of the sort of the problems. Or, let me see where. Hold on. Yeah. Okay. So more recent recognition of sort of the challenges of estimating individual inbreeding with pedigrees, along with the recent advancement in genomic technologies, has really made people interested again in the question of how we should measure individual inbreeding. And the pedigree inbreeding coefficient for a long time has been the standard measure of individual inbreeding. Josephine Pemberton wrote these papers in the early 2000s, basically saying we need to use pedigrees. Pedigrees are the way to estimate individual inbreeding. But this is interesting because we've known for a long time that the pedigree inbreeding coefficient does not equal the realized proportion of the genome that's identical by descent. Fisher pointed this out a long time ago, as early as the 40s. And if we can't predict individual inbreeding highly precisely with pedigree F, then because there are tons and tons of studies that have been studied inbreeding depression using pedigrees, we may need to rethink how we should do this. The effects of inbreeding may be greater than we think, and they might be more frequent than we think. So several studies in the early 2000s evaluated the precision of marker-based measures of individual inbreeding by taking the approach of basically estimating the correlation between multiple locus heterozygosity and the pedigree inbreeding coefficient. And this approach assumed that the pedigree inbreeding coefficient was a good measure of individual inbreeding. And unanimously, it was found that heterozygosity at a couple dozen microsatellites up to several hundred SNPs was pretty weakly correlated with the pedigree inbreeding coefficient, with the inference made that heterozygosity is a poor predictor of individual inbreeding. But the problem with this is that F sub P, the pedigree inbreeding coefficient, isn't really the parameter we're interested in. It's the realized level of inbreeding of genome. And really the first study to use simulations to look at this, to try to determine whether a pedigree or molecular measures of individual inbreeding were more precise, was Matt Keller and others published a study in Genetics in 2011, and they simulated populations meant to represent humans, using effective sizes between 100 and 10,000. They assumed there are only 2 chromosomes in the genome, however, and that the map length of the genome was 220 centimorgans. And to evaluate the precision of these measures of individual inbreeding, they estimated a correlation between what they considered as a proxy for realized genomic inbreeding, and that is the number of homozygous rare alleles within an individual. The correlation of that with the pedigree inbreeding coefficient and 3 marker-based measures of individual inbreeding. This is F rho, as I described earlier. These are 2 heterozygosity-based measures of individual inbreeding. And they found that the genetic markers, this is using sequence-level data, were much more strongly correlated with the number of homozygous rare alleles in an individual than was the pedigree inbreeding coefficient, suggesting that pedigrees were poorly performing in this study. One challenge in this study, though, is that the number of chromosomes is very small, and that the map length of the genome is very small. So that means the noise, basically, in the pedigree inbreeding coefficient is really, really high because the recombination rate across the genome is so low. So Fred and I and Gordon, as part of my PhD, decided to do a similar study looking at small populations and using genomic characteristics that are more realistic for wild populations of mammals. So I simulated small random mating populations, assuming that I simulated two different demographies. One was simulations of partially isolated populations with a small effective size of 75 and one immigrant per generation for 150 generations. Then I simulated a large population of an NE of 500, relatively large, for 90 generations, and then threw in a bottleneck at the end for the last 10 generations. So this is a recently reduced or bottleneck population. And the genomes were 3 gigabase genomes, 20 chromosomes, a 3,600 centimorgan genome, and a reasonable mutation rate for mammals. So this accounts for the stochastic effects of linkage and recombination, I think in a pretty realistic way. And these are the results of that study. So the results for the pedigree inbreeding coefficient are over here, so please concentrate over here for a moment. On the y-axis is the r squared from a linear regression of pedigree inbreeding coefficient versus f, the realized individual inbreeding. And on the x-axis is the number of pedigree generations, so the amount of pedigree information that was used. And a typical number of generations in a pedigree for a wild population is, say, between 3 and 10, with a few studies getting up to this many generations. So we have a relatively imprecise measure of, it's a relatively imprecise measure of individual inbreeding. And on the right, we have similar results for 3 marker-based measures of individual inbreeding. This is just a heterozygosity-based measure of individual inbreeding. This is a measure of individual inbreeding that uses a hidden Markov model to identify runs of homozygosity. And this is F rho estimated with the program Plink. And with about 5,000 markers, the heterozygosity-based measure of individual inbreeding had higher precision in this demographic scenario than even a pedigree including 20 generations. And the F rho measure of individual inbreeding required a lot more loci, up to around 25 or 35,000 markers to become a precise measure of individual inbreeding. So I should say the bars are the standard deviation among 20 simulation replicates. So we simulated a recently bottleneck population. We see similar results for the pedigree inbreeding coefficient, except the number of generations doesn't really matter as much. Flattens out, goes up more quickly and flattens out. And for a few of these measures, you need fewer loci to have really precise measures of individual inbreeding. So in these cases, between 5 and 10,000 markers were enough to really precisely estimate realized genomic inbreeding. And so some people have used empirical approaches to test this also. So if you have a natural population, you have fitness data on individuals, you can test for inbreeding depression using a pedigree and using genomic measures of individual inbreeding. And in these 4 studies, there's stronger statistical support for inbreeding depression, and in some cases, evidence of stronger inbreeding depression estimated from genetic markers and from the pedigree, which suggests that the genomic measures are providing a better measure of individual inbreeding in these cases. So we're getting close to the end. So what does genomics mean in the study of inbreeding depression? I think it's pretty obvious that when you have resequencing data, you can really easily, very precisely estimate individual inbreeding. And to identify runs of homozygosity, you really need a chromosome level assembly, which are still pretty rare for natural populations, but as Jeff mentioned, becoming rarer. And marker-based measures of individual inbreeding based on large amounts of data, when you have thousands and thousands of loci, you can really precisely estimate individual inbreeding. And I should say, these are sort of rough numbers for how many are needed for different measures of individual inbreeding, but they're very rough. and that should this should vary from the number of loci needed to precisely estimate individual inbreeding will vary from study system to study system, depending on things like the genome size, the variance in realized individual inbreeding among individuals, and the historical demography, which affects the length distribution of runs of homozygosity. So basically, the empirical results that have been published and the simulation results all suggest that we should really concentrate on using genomic measures of individual inbreeding, either instead of using pedigrees or in addition to using pedigrees. We should be able to more precisely estimate the effects of inbreeding on fitness in natural populations using molecular measures, and it will also allow us to begin to try to map loci associated with inbreeding depression. And we can begin to ask questions like, or to begin to more thoroughly answer questions like, how strong is inbreeding depression? What are the effects on population growth of inbreeding depression? Which loci are involved? How many? What are the effect sizes? And we can use approaches like something called homozygosity mapping, and also GWAS approaches that are based on runs of homozygosity to do this. And I just want to talk about a few examples of this briefly. So homozygosity mapping is an approach that's used to detect loci that are causing inbreeding on really simple traits, like really obvious Mendelian recessive traits. And studies that use homozygosity mapping are basically use a case control design, where we have individuals that have something and individuals that don't have something. For example, individuals that have a disease or don't have a disease. And then we identify candidate regions for loci responsible for this, where all affected individuals are identical by descent for the same exact haplotype, and none of the unaffected individuals are homozygous for that haplotype. This is an example of an analysis of homozygosity mapping in cattle. So they were looking for the locus responsible for this disease, congenital muscular dystonia. They used a high-density SNP chip to do this analysis. So they had some control individuals, these are unaffected individuals, and they had some case individuals. Don't pay attention to the red and yellow. Basically, they looked at the genome and identified this chromosome as being important. So the parts of this chromosome in the cases they identified IBD segments where the gray and black bars are. And every affected individual was IBD for this chunk of this chromosome for the same haplotype. And subsequent Sanger sequencing of this segment in a lot more affected individuals and a lot more unaffected individuals showed that 81 individuals were homozygous for the same missense mutation in this ATP2A1 gene, which is involved in fast twitch muscle development. So suggesting that they found the mutation responsible for inbreeding depression in cattle for this trait. I think this is a really cool approach, and it could be used in some natural populations or captive populations of conservation concern, where we have an obvious recessive Mendelian trait that's causing something awful. And when you have a more complex trait, something that's caused by or affected by variation in a large number of loci, you have to use a different approach, using more of a classical genome-wide association study approach. And basically what GWAS, you can use runs of homozygosity in a GWAS by scanning the genome for either using windows or SNPs and looking for an association between the presence of a run of homozygosity in a part of the genome and a phenotypic trait. So you're looking for a correlation between presence of run of homozygosity in a part of the genome and variation in the phenotype. This is one study in humans that looked at schizophrenia using this approach, and what they did was break the genome up into 500 kb windows, and they did a logistic regression of schizophrenia status on whether the window overlaps with any runs of homozygosity. They failed to find anything, but this is an approach that's been used. It's a sort of rough approach. And then this study, Price et al. Used in cattle a SNP by SNP test And so they basically scanned the genome for each SNP, scored whether that SNP overlapped with a run of homozygosity, and used that as a predictor variable in a linear mixed effect model, looking for loci contributing to inbreeding depression on milk fat and protein yield. So these are approaches that could be used in natural populations. I should say that the power of approaches like this might be quite low, except for in very few circumstances where you have really large effective loci contributing to inbreeding depression. And it'll be really interesting to see if those pop up in natural populations, and I'm somewhat skeptical about our ability to identify the mutations causing inbreeding depression on complex traits in natural populations. So when we have genomic data, if we're interested in studying inbreeding depression, when do we need mapping information or a really large number of loci? I think that's an important question to think about for you guys if you're interested in inbreeding depression. So basically, precisely identifying runs of homozygosity both requires lots of loci, a really large number of loci, hundreds of thousands preferably, and mapping information. You need to know where loci are in the genome, and you need a lot of them to identify identical by descent chromosome segments explicitly. And basically the usefulness of these is that you can explicitly estimate F, which you can't do with just multiple locus heterozygosity. And you can also use these to try to map loci contributing to inbreeding depression. And as we did with the collared flycatchers, you can try to use estimates of identity by descent to learn about historical demography. And you need both large numbers of loci and mapping information to do this. So when do we not need large numbers of mapped loci? Simply detecting inbreeding depression does not require mapped loci or genome resequencing. Essentially, if you have thousands and thousands of loci, even if they're unmapped, multiple locus heterozygosity calculated over that many loci is a really precise estimate of genome-wide heterozygosity, which is an indirect measure of individual inbreeding. And you need far fewer loci than you need when you're trying to detect runs of homozygosity. And if you're interested in just detecting, if you're just asking the question, is my population inbred, and is inbreeding depression likely, basically all you need is estimates of heterozygosity in that population and other populations. If you have a population that has lower heterozygosity than other populations, those individuals in that population are more inbred. Basic population genetics theory tells us that, and you don't need many loci to do that. A couple hundred SNPs typed in 30 or 40 individuals is plenty to detect, to estimate genetic variation within populations, and to sort of have relative estimates of population level inbreeding among populations. Thanks a lot. Questions? I'd be happy to answer. Yeah, Paul?
So another use of pedigrees is to estimate heritability if you have genetic condition. Does the same argument apply that basically lots of genetic markers is better than a pedigree if you want to estimate heritability?
So you have, yeah, so in, a lot of the quantitative genetics literature now, we're sort of, looking at, I'm not a quantitative geneticist, I've dabbled in this. But, you can build a genomic relatedness matrix. So you have, you can estimate pairwise relatedness among individuals using large numbers of SNPs. And that, you're basically doing the same thing there as when you're estimating individual inbreeding. You're just estimating the similarity of two different individuals' genomes instead of the similarity of two copies of the genome within an individual. And you can, look at, well, a lot of the approaches in quantitative genetics now are looking at similarities in phenotype as a function of similarities in the genome to estimate heritability. So, I'm not an expert on how well those work, but yeah, you can use genomic data on how similar individuals are genomically.
order to, because given that a lot of populations can persist under some level of inbreeding, it seems really important to know if you're a small population that you're worried about actually experiencing inbreeding depression. And so, I mean, I think you said this at some point, like the ability to pair these genomic data sets with pedigree is like probably the most powerful, but yeah, it's not really a question, it's just.
Yeah, so knowing, having fitness, and so I really think the role of long-term studies is really important, because without long-term studies, you don't have things like lifetime reproductive success and lots of fitness data over individuals for many generations. And by virtue of collecting those data, you have a pedigree. And I still think that long-term studies are the most important. It's really hard to go out into a population and measure some fitness component over 1 or 2 generations and sort of feel like you have a good sense of what's going on in a population. Whereas if you follow life history over 30 or 40 years, as has been done in a handful of long-term studies, it's really valuable.
Going back to where you were inferring historical demography, would it be possible to narrow your confidence interval if you have historical samples or ancient DNA available, or can those be used in that sort of algorithm?
Perhaps. Yeah, I'm not sure. So, yeah, you just have sort of a different starting point. So, for example, yeah, so if you if we had samples, we have samples from here. If you had samples from here, with the same number of individuals, that would be interesting to do that. One thing we're doing is, so that we think we know what the source population for this population is. It's a different island in the Baltic, this island called Gotland, and we have some both contemporary and historical samples from that population, and an idea is to estimate NE using samples, historical and contemporary samples from Gotland. And I'll be interested to see if it's about this big, because I think this is an estimate of NE for the source population. So it would be cool to do that. And I think it would help, definitely. Thanks a lot.
I'll take a comment, and then you had a hands-on use link.
Yes, I do.
To get started on.
Yeah, we only have 25 minutes, and I'm not organized.
Right. We could start it after lunch then, or
Yeah, either
talk
after
about
lunch,
it
or
then?
even better this evening.
Oh, okay.
Just, I mean, it's very short, so
Okay.
it shouldn't take long, but we can do it this evening.
Yeah, okay, that'll work, that'll be fun. We'll send that around. I was just gonna loop back to the comment that followed from this topic after Fred's talk, that it's likely because most of the vast publications in the literature that tests for inbreeding depression and detected only used a handful of markers or pedigrees. Our understanding from the literature then suggests, with this kind of new information, that we have that inbreeding depression is far more common, widespread, and severe than our current understanding from scientific literature. And that has obvious implications for conservation. So I think that's a big deal.
I got a question, like following up on that, of so many captive breeding programs are very, pedigree based. Is there a push to incorporate this type of identification of inbreeding now in the kind of captive population management?
Yeah, I have a colleague who's just doing a postdoc working on Galapagos tortoises, trying to use RAD data in Galapagos tortoises to inform captive breeding programs. So, if genomic estimates of pairwise relatedness are better than pedigree-based estimates, it should help.
So it seems to me that aside from the flycatcher case, you have species, whether they're fish, insects, whatever, that would be invasive, that are common or currently invading. And there may be a relationship or a way to look at whether species that invade that maintain low problems, low growth, and those that really become invasive, looking at some kind of relationship between this homozygosity, whether they actually, because they go through that founder effect, have inbreeding depression phenomena that go on that keeps them low versus those that don't have any of that. Are you aware of any cases where we've kind of looked at the invasive species side of this?
No, I'm not an invasive species person, but it would be interesting. So basically to look at how the strength of inbreeding depression predicts invasion success.
Yeah.
You know.
I don't know, I'm just thinking that might not always be the best predictor. I mean, you've got the Argentine ant as an example, and the reason why it's such a good invader is because they're all so genetically similar. They all get along real well, and so they make these gigantic super colonies, and they don't fight each other. So it may not always be a predictor.
So they may have been purged of any.
They
inbreeding.
recognize each other as
Yeah.
nestmates because they
Yeah.
lost genetic diversity and recognition systems.
So have you calculated your inbreeding coefficient?
Mine?
Yeah. Your comment earlier.
No, I was kidding.